It is my pleasure to introduce the second speaker in our third session of the day, Dr. Johanna Lampe. Dr. Lampe is a professor and associate division director in the Public Health Sciences Division at Fred Hutchinson Cancer Research Center, so our neighbors here in Seattle, and a research professor in the Department of Epidemiology at the University of Washington. She received her PhD in nutritional sciences with a minor in biochemistry from the University of Minnesota, and trained as a postdoctoral fellow in epidemiology, also at the University of Minnesota, before joining the faculty at Fred Hutch. Dr. Lampe's research focuses on the effect of diet constituents on cancer susceptibility in humans, and the effects of human genetic and gut microbial variation on response to diet. Her collaborative studies have involved the application of a variety of omics approaches to characterization of biochemical and physiological response in controlled feeding studies, and to the development of biomarkers of dietary exposure. She has received many awards for her work, including the American Society for Nutrition Mary Schwartz Rose Senior Investigator Award for research on the safety and efficacy of bioactive compounds for human health, and the National Cancer Institute's Division of Cancer Prevention Stars in Nutrition and Cancer Award, which recognizes research contributions in the field of nutrition and cancer. Today, Dr. Lampe will talk about her work involving diet-gut microbiome interactions and influence on controlled feeding studies and metabolic phenotype. So please join me in welcoming Dr. Lampe.
Great, thanks so much, Tomas, and it's certainly a great pleasure to be a part of this today. Many thanks to ISB for organizing this, and to all of the speakers who've come before me with some just amazing studies and results. It's really been exciting. So what I'd like to do today is to take a diet-centric view of the microbiome diet interactions, and to talk about some of the work that we've been doing and some of the approaches that we've been using. So from the standpoint of studying diet, one of our big challenges is that the human diet is extremely complex, and there are thousands of compounds that are part of it. And when we think about how we study diet and also how we eat our diets, foods are very complex mixtures, and we often assess them by looking at individual compounds or individual foods and classes, but we really are eating something more along the lines of dietary patterns and the totality of diet. And the whole field of nutrition is actually coming to embrace this more and more, as we recognize that although we've taken a very reductionist approach in the past, following one nutrient, one compound, that really, because of the interactions between all these dietary constituents, we have to think more broadly. In certain populations also, dietary supplements are a huge component of dietary intake, and so we certainly can't ignore those either. So today we've heard a number of different research approaches for studying your gut microbiome, and certainly this comes into play in relation to the diet-gut microbiome interactions too. We've heard a lot in relation to a lot of the in vitro and even the in silico bits before that, as well as the animal models. And the piece that I typically fit in is in the area of small-scale controlled interventions, what is often termed human mechanistic studies these days, to contrast that with clinical trials, which often are related to understanding a disease outcome or a therapeutic outcome. Here we're really just trying to understand the biology, and from the standpoint of the kinds of things that we can ask, certainly mechanistic questions, focusing on particular nutrients, compounds, as well as the microbiome variation. And then on the other end of the spectrum, when we start to get into large population-based cohorts, where we rely more on observational studies, we're really looking more at the broad systemic effects, food and diet, and again, but we can still apply the variation in the microbiome to these studies. So many of the randomized clinical trials would fall into this individual and community-based interventions, which tend to be less invasive on study participants. A lot of what we do in our controlled feeding studies and the human mechanistic studies put a lot of demand on our study participants. So just going back to general gut microbial metabolism, I think it's always good to remind ourselves of where this is happening and what the impact of the anatomy of the human host is on what the microbes see. Thinking about it from the standpoint of how we're designed when we eat food, really human digestion happens before any of the microbial digestion occurs. So a lot of the enzymatic releases within the small intestine are handling a lot of the macronutrients, carbohydrate, protein, and fat. And so it's really the leftovers that become the substrate for the gut microbiome. I mean, certainly down here in the lower part of the small intestine, you do have microbes, but the major action certainly is within the colon. So thinking about it from that standpoint, the microbial substrates are really the components of our diets that we can't digest and absorb and end up and down in the lower gut. And as Thomas Gurry pointed out, you know, that's highly variable. If you're eating a huge load of protein, you may find that there are actually a lot of proteins and peptides that end up in the colon that become substrate for amino acid fermentation. But it really means then that a lot of the reactions that are carried out by the gut microbes relate to reactions that we don't have the enzymes to carry out. So we've talked about fermentation already today, denitrification, sulfate reduction, capacity to break a lot of the ring structures, for example, in relation to a lot of the polyphenol compounds in plant foods, as well as a lot of hydrolysis and deconjugation reactions. So these are sort of the mainstay of a lot of the microbial action. The reason I got interested in this was the National Cancer Institute started an initiative, almost 30 years ago, to understand better the impact of a number of dietary components that aren't considered to be essential nutrients, but may actually affect risk for cancer. And there were a number of phytochemicals that were recognized as their potential role-playing effect. And it became apparent that many of these compounds actually are heavily metabolized by the gut microbiome, such that exposure to them can vary highly across individuals. And it was the lignans that really caught our eye from the standpoint of these being associated with high fiber foods, and we were interested in pursuing this area. At the time, my group was very interested in conducting controlled feeding studies to look at effects of diet on various biomarkers of cancer. But as we recognized, even when we controlled the diet really well, there was a huge amount of variation in response. And we started out by looking at host genotype diet interactions, and then really realized, particularly in relation to the phytochemicals, that it's really the host microbe diet interactions that probably warrant more attention for a lot of these effects. In the context of the plant lignans, there's been a substantial amount of work that has been done to understand their impact in relation to chronic disease. A lot of prospective cohort studies, case control studies of colon and breast cancer, a number of cardiovascular diseases, suggesting a reduced risk for these diseases. Experimental human studies showing changes in things like hormone metabolism, inflammation, and concomitantly with this, experimental animal studies, again, supporting both the effects on these mechanisms, but also the outcomes in relation to tumorigenesis. So lignans can act through a variety of different pathways. This is the case with many phytochemicals when you come to study them, unlike certain drugs that are often targeted for a particular pathway. We find that many phytochemicals, and lignans in particular, can interact through a variety of different receptors. And in some cases, they're acting as agonists, for example, through the NRF2 KEAP1 pathway, affecting a number of the antioxidant enzymes and detoxification systems, biotransformation. Also, through estrogen receptor mediation, PPAR gamma, and the RXR receptors. And also they can act as antagonists and compete for interaction with some of the other receptors, and similarly, consequently affect a wide range of processes in the human body. So when we look at where these compounds are found, these are diphenolic compounds. There are a number of different flavors of them and different compounds themselves. Many of them are present as glycosides, and they come from plant foods, predominantly those that are very high in dietary fiber. So a lot of the whole grains, nuts, seeds, fruits, and legumes. And so it's really this component of what we're feeding in the context of a diet, but what actually is going on. And that is related to the gut microbial metabolism of these compounds to two major products, Enterodiol and Enterolactone. This is starting with secoisolariciresinol diglycoside and metaresinol glucoside, and they undergo a number of enzymatic steps, including hydrolysis of the glucosides, demethylation, dehydroxylation, to produce Enterodiol. And it's real, Enterodiol has been the compound of most interest because of its bioactivity and potential effects on health. But in some of the initial microbiome work that's been done, they've identified some bacteria that can carry out all of these reactions. But for the most part, this requires a consortium of bacteria to allow for the production of enterolactone. And in a study a while ago, but which actually shows really nicely, is just the variation in how individuals' bacteria are producing the end products. You have some, this is a pharmacokinetics study where individuals were given secoisolariciresinol diglycoside, or SDG as we call it. And you see that this person produces copious amounts of enterolactone with some Enterodiol. You've got some people who aren't really making much of either of them, again, all based on the same dosing of substrate. Again, somewhere it's more enterolactone versus Enterodiol, and somewhere who aren't making any enterolactone but are producing quite a bit of Enterodiol. So again, really dependent on the microbial community as to what the end products are. So we were interested in embarking on a study to look at the potential effects of enterolactone on a number of cell signaling pathways in the colon pertinent to colorectal cancer risk. And started by selecting a plant lignan supplement to allow for conversion to the enterolactones, enterolactone and Enterodiol, and then to look at downstream effects in the human host as a result of this intervention. And this study, which was called the Flax Effect Study, was a randomized placebo-controlled crossover intervention in 40-some healthy men and women, relatively young population. Our goal here was really just to understand the basic biology of response in the context of healthy individuals. And we provided a dose of 50 milligrams per day of SDG for 2 months. And the major outcomes were gene expression in colon and rectal biopsies and exfoliated cells. We had participants come in for sigmoidoscopy, and we would take colon and rectal biopsies at the end of each intervention, 2-month intervention, and then stool samples for microbiome interrogation. We also measured 24-hour urines to look at enterolactone excretion. So enterolactone produced in the colon is well absorbed, undergoes conjugation with glucuronide and sulfate in the liver, and is then rapidly excreted in urine. And so looking in these 24-hour urines in the context of the squares, which was the lignan supplement, and the placebo in the triangles, we set this up so we went from lowest excretion to highest. And as you can see, there's a wide range in capacity of individuals to produce enterolactone. Some individuals on the supplement are not producing any enterolactone at all. Others are producing lots of it. And even on, you see this too in the context of the placebo, where people are exposed to some lignans as part of their usual diet, but even then the amounts are relatively low. So it was based on these differences that we actually split a lot of our analyses between those individuals who fell at the bottom half of ENL excretion and those who were in the top half for many of our comparisons, and I'll share some of those data. So this is looking at gut microbial response to the supplementation. We collected stool samples at the end of each of the 2-month intervention periods, and this was 16S analysis, so pretty much at the general level, looking at the community at the end of the 2 time points. The placebo is the circle and the lignan period is the star. And we didn't hypothesize we would actually see a shift in the gut microbial community as a result of the intervention. This is 50 milligrams of SDG is a relatively small perturbation in the context of habitual diet and substantial fiber intake. So we weren't really expecting much of a shift, but you do see that there are some shifts within an individual when they are on the 2 treatments. And what's more interesting in the context of this is also overlaid with that is their urinary ENL excretion from dark blue being very low excretors to the yellows being high excretors, and that the overall communities tend to differ between the low and high excretors. So you have a lot more of the yellows and light greens over here and the darker ones over here. That's certainly a lot of overlap, which isn't too unusual in human studies. The other thing that we did then was to look at colonic mucosal gene expression using RNA-seq in the biopsies from these individuals. And although we didn't really see a major impact of the intervention itself on changes in gene expression with the SDG supplement, what we did see is that when you look at differences in expression between the low and high ENL excretors, there are a number of things that come up. Many of them relate to inflammation. For example, number of anti-inflammatory upstream regulators, including TGF-beta1 and IL-10, were suppressed in the low ENL excretors. And similarly, using the fecal exfoliome, as we call it, and this is actually epithelial cells sloughed into the stool that you can actually use RNA-seq on a sample here. This is work of Rob Chapkin at Texas A&M, who has established this method, which is really nice from the standpoint of not, in the future, not having to go in and take biopsies, but rather just rely on the stool samples to be able to look at what's going on to the gut epithelium. And in this case, we find, again, comparing the low and high ENL excretors, that we see an upregulation of NF-kappa B and NOS2 among the low excretors, suggesting that there's a differential effect of having the capacity to produce ENL versus not. And it doesn't necessarily mean that it's the ENL that is doing it, but it's really the microbial community associated with that phenotype. So this is work that we're following up on with a goal to try to understand this better. As I indicated, plant lignans are really an integral part of high fiber foods. And so we start to think about the totality of diet and the combination of both the high fiber foods as well as the rest of the diet. We've taken to designing studies that actually allow us to look more at dietary patterns rather than specifically focusing on individual foods. This is a study that was conducted to design 2 different diet patterns, one that was high glycemic load and one that was low glycemic load. The low glycemic load diet, which the idea is that you're picking foods that result in a low area under the curve for glucose response as a result of feeding. These tend to be foods that are high in whole grains, legumes, fruits, veg, and tend to be low in refined grains, added sugars. And in the literature, we know both from the epidemiologic literature as well as a number of interventions that diets that tend to be higher in these foods, and this is, as a result is why a lot of the public health recommendations focus on encouraging higher intake of these, is that they're associated with the reduced risk of a number of chronic diseases through effects on inflammation, metabolic regulation, etc. Whereas a diet that's high in refined grains, added sugars, and more high glycemic load foods are associated with metabolic dysregulation. And a number of studies had suggested that these high glycemic load diet may also be contributing to higher risk of several cancers. So it was this that set us out to do a controlled feeding study to examine this. And we're very fortunate at Fred Hutch that we have a shared resource that provides us with a facility to conduct interventions in humans. We have the Human Nutrition Lab, which is essentially a commercial kitchen and dining area where people can come in and eat on site, and we can provide them with all their food for the span of the study. As you might imagine, during COVID, things are a little different. Nobody's in the dining room, but we are delivering food to participants to be able to allow some of these interventions to continue. Similarly, this facility has a exercise lab, which allows for exercise interventions too. So what these controlled feeding studies allow us to do is to control the diet for the time period that the participants are involved in this. And so for this high and low glycemic load diet, this was the approach we took. The one thing, of course, a study like this has its limitations, and certainly you can't carry on an intervention sufficiently long to allow for disease outcome as an endpoint. And so you're really relying on intermediate biomarkers to monitor the effects of the intervention. What a controlled feeding study allows you to do is to control both what you're intervening with, but also allows you to control the background diet. So often in nutrition, you think, okay, I'm going to give somebody this food, but giving them that food without taking into consideration the context in which they're consuming it, i.e. what other things are they eating, may actually not give you the information that you're trying to obtain from your intervention. So this approach where you can really control diet to the max helps. You can design these diets to test dose response, and then typically the intermediate markers of disease susceptibility are your outcome measures. Also useful from the standpoint of developing markers of dietary exposure, where you can come to understand, okay, you feed somebody a diet high in cruciferous vegetables, i.e. broccoli, cauliflower, cabbage. What does their metabolic profile look like from the standpoint of the kinds of compounds that they're producing, things showing up in their urine, things showing up in their blood. So useful for that too. So in the case of the study that we conducted of low and high glycemic load diet patterns, this again was a randomized crossover design. To the extent that we can, it's often really nice to have individuals serve as their own control compared to giving one group of people one diet and one group the other, particularly in the area of the microbiome where there is so much heterogeneity. Understanding how something works within an individual is really important. So taking this tact has been our usual approach. Again, healthy individuals, 18 to 45. And the diets were designed to provide same number of calories per day for an individual on both the high and low glycemic load diets. Similar macronutrient distribution, that is 50% calories from carb, 15 from protein, remainder from fat. And then as a result, selecting the carbohydrate containing foods, you're really looking at differences in carbohydrate quality, also substantial differences in fiber content as a result of picking foods that are low glycemic load, which tend to be higher in fiber. They also tend to be higher in plant lignans compared to a high GL diet, which low in fiber, but actually it's actually substantially higher than the usual fiber intake of the US population. I think our fiber intake is estimated at a mean value of about 15 grams per day. 28 grams per day is actually well within the recommended range, and lower amount of lignans. And then the main study was actually designed to look at effects on metabolic and inflammation biomarkers that had been associated with cancer risk, just to show you that the diet actually did have an effect with regards to glycemic response. The open squares are the low glycemic diet, so i.e. glucose area under the curve is lower with a low GL diet versus the high GL diet. And similarly, insulin response is lower on the low GL diet. So we followed this up with additional study, given that we had samples collected as part of this, to examine the responses to the diet in relation to taking a proteomic, metabolomic, and microbiome approach. And so we had bloods collected beginning and end of each 28-day feeding period. This was a crossover design, so after a 1-month washout in between, the individual received the diet they didn't get in the first part. So again, the contrasts were within an individual. And looking at the proteomic response, we see a number of different proteins differentially expressed as a result of the low GL versus high GL diet. In this case, these are proteins that were expressed. This was using an antibody array approach. So a number of proteins that were lower on the high GL diet here, and then the red is higher on the LGL diet. Number of pathways relevant to DNA repair, replication, post-translational protein modification. Interestingly, we actually recruited the participants in the study on the basis of percent body fat, and there were a number of responses that differed based on whether or not an individual had a high fat mass versus low mass. As you see here across the two columns, in some cases the responses are similar, but for some of these they differ depending on percent adiposity. So in relation to the metabolomic analysis, and I think this is from a microbiome standpoint more interesting even, is that we saw a number of metabolites that differed by diet. This was a targeted metabolomic approach, relatively small number of metabolites that we were monitoring, but we identified several pathways that differed between the two diets, including tryptophan metabolism, branched chain amino acid metabolism, and trimethylamine production, all of which decreased in the context of the low glycemic load diets. And looking at these responses in the context of C-reactive protein, a measure of inflammation, we see too that many of the concomitant changes in both these metabolite pathways as well as CRP were going in a similar direction. Again, not necessarily causal, but providing us a way to move forward on interrogating these further. One of our questions was, given that a number of the metabolites that we were identifying in relation to these outcomes are microbial metabolites, what were the effects of the diets themselves, in the case on secondary bile acid production and the relationship between enterolactone production and the plasma bile acids. As we've heard today a number of times, the primary bile acids produced in the liver, cholic acid and chenodeoxycholic acid, are converted by microbes to deoxycholic acid and lithocholic acid, predominantly in humans. And these compounds have, as well as serving a purpose with regards to lipid absorption in the upper gut, really have become to be recognized as signaling molecules for a number of different conditions. So looking just at plasma bile acids using a targeted platform of 29 bile acids, we see interestingly higher plasma bile acid concentrations in relation to the low glycemic load diet. I would have hypothesized the other way around, but evidently aspects of the diet are contributing to both increases in the primary bile acids, the cholic acids, conjugated forms, as well as lithocholic acid. Now, good to keep in mind that the fat content was, the same on both these diets. So it's not necessarily that you would expect that a higher fat content causing more release and potential conversion to lithocholic acid. But again, a lot of things still to be worked out on that. In this case, in this study, when you look at lignan intake on the two diets across the individuals, you see that there's a huge range in enterolactone excretion, but it doesn't correlate with the lignan intake. So again, here, the microbial conversion is such that there is a huge range in actual production of enterolactone. And looking at enterolactone excretion in relation to those bile acids, we see that an analysis, association analysis, we see that higher enterolactone excretion is also associated with higher bile acid concentrations for a number of these bile acids. Some follow-up work that we've just done recently looking at some of the enzymes involved in enterolactone production by the gut microbes, that many of these are tracking with some of the same enzymes involved in bile acid, secondary bile acid production too. So some of these overlaps of the capacity to produce both enterolactone as well as some of the secondary bile acids likely come into play here. So just to summarize and to bring us back to where we started in the context of diet and the microbiome, coming at it from a nutritional standpoint and diet-centric view of the universe. And we think about diet and we think about why we eat. A lot of it is for fuel, as well as many other components of diet that are essential for structure and function of the host. In this day and age, certainly a lot of our diet has resulted in energy excesses, contributing to many different chronic diseases, as well as excesses in some of the dietary constituents having this effect. And I think the nice, exciting part about the discussions today is also thinking about the interaction with the gut microbiome. Like if you come at the world from a gut bacterial or gut microbial centric view, you're thinking about what is the impact of the gut bacteria on disease risk and host health. But we all have to remember to keep both of these pieces in mind, both with regards to the effect of diet on the gut bacteria, as well as the gut bacteria effect on the metabolism and exposure of the host to these. So it's an exciting time. Having started at this before any of these molecular tools were available to even interrogate the microbiome, it's really been fun to be a part of this. I'd just like to finish up by acknowledging the support from NCI that we've had over the years and the Fred Hutch, and really to thank Meredith Huller, who's a microbial ecologist who has worked with our group for some 15 years now, helping to spearhead some of the microbial work in this area, as well as Sandy Navarro, who does a lot of our metabolomics analysis in collaboration with Dan Raftery and Ali Shoje at the University of Washington. As I mentioned, Rob Chapkin has developed a lot of the exfoliome RNA-seq analyses that are allowing us to conduct more and more interventions without having to go in and take biopsies, which is simplifying life, as well as other collaborators at the Hutch and around the world. And with that, I'll stop and thank you for your attention. I'm ready to help answer questions.
Thank you, Dr. Lampe. That was a wonderful talk.